The Meet the Researchers series has been a wonderful way to engage with the community about the inspiring research that happens at IMB. And today we're looking into the future of pain relief. You'll hear directly from three of our fabulous IMB scientific staff and from our special guest, and we'll have the opportunity to ask questions and gain insight into their scientific minds. I'd like to acknowledge the following special guests we have in the audience today. Our IMB Directors Circle members, Dr. Selwyn Russell, Patricia Ash, Pauline and William North, who I believe are joining us online, Wendy and Alan Grummett, also online. We also would like to welcome Anita Fung, President of Quendo, who's tuning in online. Quendo advocates for those who are affected by endometriosis and other pelvic health-related conditions across Australia and New Zealand. And we also are privileged to welcome today Nicolette Ellis, the President of Chronic Pain Australia, who you'll be hearing from a little bit later. And now it's my pleasure to welcome Dr. Hana Starobova. Hana is a research fellow at the University of Queensland. She's a trained pharmacist and neuroscientist. Her research focuses on the understanding of cancer therapy-induced adverse effects, including late effects and the development of effective prevention strategies. Hana's long-term goal is to create a research program bringing together researchers, industry partners, and clinicians who will work together towards treatment strategies to improve the outcomes and the long-term impacts for pediatric cancer patients. Hana is an internationally recognized expert, and she was awarded the 2019 Women in Technology Award and the prestigious Children's Hospital Foundation Fellowship. Today, Hana will share how life-saving cancer therapies negatively impact pediatric cancer survivors' lives, and how she is working towards developing effective strategies to prevent the long-term impacts of cancer therapy in children. Thank you, Hana.
Thank you, Wendy. Hello, everyone. How is everyone feeling today? Well, today I was cycling to work and I was actually really freezing. Now, and the funny thing is that I'm originally from Czech Republic, from Europe, and the winters there get really cold because the temperature goes to -15, -20 degrees. Now, in 2014, when I finished my studies in Germany, I moved to Brisbane. And during the time, I actually found the temperatures here in Brisbane during winter extremely warm. So I would be running around in shorts and t-shirt. Now, 10 years later, living in Brisbane, I got perfectly acclimatized to the weather here. And now when the temperatures go below 20 degrees, and maybe you can relate to it, I'm immediately freezing and grabbing a thick pullover. Now, I have two dogs. They both also don't like cold temperatures, and their favorite thing to do at the moment is to basically lay next to the fireplace. And I'm pretty sure they would not survive a day in the European winter. Now, enough about me, and let's focus on my talk. So now my talk has the title Surviving Cancer. Now, maybe you ask yourself, what surviving cancer has even remotely to do with pain research? Now, during my talk, you will find out that many cancer patients, but especially cancer survivors, actually experience long and severe pain. But before we dig deep into research, I would like to ask you a question. Who in this room knows someone who has or had cancer? Could you please put your hand up? Okay, so almost everyone here in this room has put their hands up, and I'm pretty sure that also the audience on Zoom will probably think the same, and they will know someone who was impacted by cancer. Now, imagine the doctor tells you that your child or someone you really care for has cancer. So in this terrible moment, probably the only thing that you hope for that they survive. And yes, absolutely, survival of the cancer is very important. But surviving the cancer is only half of the battle. Now, let me tell you a true story. Ellie, who is on these pictures, was admitted to hospital in 2015 with a high fever and fatigue. Now, after several tests, Ellie was diagnosed with leukemia, which is a type of blood cancers. Now, Ellie was treated with several cycles of cancer therapy that it basically includes a combination of different chemotherapy treatments. And these days, the chance to survive cancer such as leukemia is about 90%. So Ellie had a really good chance to survive the cancer. However, at some time in her cancer therapy, Ellie has started experiencing severe pain in her hands and feet. She became severely constipated and fatigued, and she has stopped the hearing on her left ear. Additionally, she has developed neutropenia, which is basically a decrease of specific immune cells called neutrophils, increasing her chances to basically become infections. Now, due to these adverse effects, the doctors had to change the cancer treatments, and they have basically changed to less toxic, but also less effective cancer treatments. And this decision was very important because Ellie was really suffering, but it also decreased her chance to survive cancer. Now, Ellie grew into a young lady, and we are all really grateful because she's cancer-free. However, Ellie still experiences severe pain in her hands and feet. She has developed motor impairment, and she has difficulties to walk, to run, and to perform fine motoric tasks, such as playing violin, and she has frequent headaches. And all these factors basically decrease her quality of life. And Ellie is not alone, because each year there are about 1,000 Australian children that are diagnosed with cancer. And 80% of these children will develop severe adverse effects of their cancer therapy. And from these children that are diagnosed with cancer and experience severe adverse effects, about 70% will experience long-term impacts of the cancer therapy. And these impacts include pain, constipation, cognitive impairment, and, for example, speech impairment. Now, currently, there are no treatments that would either prevent or treat adverse effects of cancer therapy. The only way we actually can help these patients a little is to use physiotherapy, speech therapy, or cognitive remediation therapy. Now, you can also imagine that this greatly impacts the whole family, because often the primary carer of the child will have to reduce their working hours to be able to sufficiently support the child during this time. And of course, you can imagine that this then impacts also the family budget, which is then additional burden to all the costs the family has due to the high hospital bills. And often these families also have to travel long distances to be able to even access hospitals and type of these therapies. Now, I have already mentioned that there are actually no available treatments that would either prevent or treat cancer therapy-induced adverse effects. But why is this the case? Well, we actually know that cancer therapies, they can actually activate specific immune cells that then release substances or factors into the bloodstream. And these substances then basically damage nerves of the peripheral and central nervous system. And this can also reprogram the immune system, which altogether basically leads to development of adverse effects, including severe and long-lasting pain. But the development of adverse effects is a very complex process, and it also includes many different factors that contribute to the development. So one of these factors is, for example, gender. For certain types of pain, such as migraine and headaches, we know that, for example, female patients suffer more than male patients, and there are studies showing that this is also the case for adverse effects. The second factor is the type of cancer that the patient was diagnosed with. Now, we know that cancer cells itself can release factors that can contribute to development of adverse effects. Another factor is the patient's lifestyle, including the diet and exercise. We know that patients that, for example, exercise before, during, and after the cancer therapy, these patients actually recover much faster from neuropathies and motor impairment. Now, another factor that plays a role is the type of the cancer therapy, because not all chemotherapy treatments cause the same type of adverse effects. For example, vincristine is a drug that is used to treat blood cancers in children. And this type of drugs predominantly causes motor impairment. Now, other types of drugs are called platinum derivatives. These drugs are used to treat, for example, breast cancers and pancreatic cancers in adults. And this type of treatments, they cause predominantly pain. Now, another important factor is the age of the patient and the developmental stage of the central nervous system, of the peripheral nervous system, and of the immune system, because this also contributes to development of adverse effects. And lastly, genetic predispositions. We, for example, know that Caucasian children actually develop adverse effects of cancer therapy more likely compared to, for example, Hispanic children, which has something to do how the cancer treatments are processed and eliminated through the liver. Now, as you see, the factors that underlie adverse effects are very complex and the mechanisms are very complicated, making it for us researchers, but also for clinicians, very difficult to understand and also treat. So for this reason, we have put together a team comprised of world-leading pain researchers, cancer researchers, oncologists, and industry partners, to basically solve this problem and to develop treatments for cancer therapy-induced adverse effects. And as you see, our collaborations go beyond Australia because we collaborate with researchers based at the Columbia University in New York, and also with researchers at the University of Texas in Dallas, and with industry partners that are based in Sweden and in Switzerland. Now, one reason why there are still no treatments for cancer therapy-induced adverse effects is that most treatments that are developed to treat, for example, pain or neuropathies, and then tested in clinical studies, most of these treatments will fail to show any positive effects in clinical studies. And our team was been basically pondering, why is this the case? And basically, we have found out that at the moment, the preclinical studies that we are performing currently before we move on to clinical studies with patients, these preclinical studies actually do not take into account all these different factors and complexities that basically underlie development of adverse effects. So in the future, our team will be very focusing on developing preclinical models that will take into account, for example, age, gender, the type of cancer treatment, and also the genetic predisposition. So we can basically test effective treatment strategies in really appropriate models so that we can basically accelerate the translation into clinical studies. Now, for our preclinical testing, we use rodents. And I know this is a very sensitive topic, but I would like you to understand that if there would be no preclinical testing in animals, there will be no cure, because every treatment that we use has to be tested in preclinical models in animals before we can actually move on to clinical studies. So we as researchers in Australia, we have to show that the treatments or the drugs that we are testing are effective in animal models and are safe before we can move on to clinical studies in humans. But we are also, of course, following strict laws such as the Animal Care and Protection Act or the Australian Code for the Care and Use of Animals for Scientific Purposes to assure the welfare of these animals during the experiment. And we are working very closely with the Animal Ethics and Welfare Committee, which basically consists of members of the public and veterinarians. Now, most funding and research efforts focus on understanding cancer and on curing cancer. So we have been really struggling to find to basically receive funding from large funding bodies such as the National Health and Research Council. So our research would not be possible without philanthropic funding and without the support of the Children's Hospital Foundation, the Kids Cancer Project, and also of the US-based Prep for Gold Foundation that has been supporting our efforts for a very long time. So basically, we are always in the need for funding. And the only thing that basically holds us back from developing effective treatments for cancer therapy-induced adverse effects is simply because we do not have enough money to perform the research and to perform the clinical studies. So today, I would like you to remember a few things. Firstly, surviving cancer is only half of the battle because cancer treatments cause severe adverse effects such as long-lasting pain that significantly impact the quality of life of cancer survivors. Secondly, there are no effective treatments for cancer therapy-induced adverse effects. And one of the reasons for this is that most researchers are currently using very simplified preclinical models that do not take into account the complexity of cancer therapy-induced adverse effects as we can see them in patients. And lastly, I would like you to remember that our research focuses on developing new effective treatment strategies that would prevent adverse effects in patients so that we can make sure that children like Ellie not only survive cancer, but can also live long and fulfilling lives without pain. Thank you for your attention, and it was all from me.
Thank you, Hannah, and I really appreciate you sharing some of the challenges that you face for this important research. And we'll have time for questions at the end, so please do keep in mind anything you'd like to ask. I'd like to now welcome Dr. Sam Robinson, who's an expert on plants and animals that sting, and the biology, chemistry, pharmacology, and pathophysiology underlying those stings. His research is providing new understanding on the mechanisms of chemical defense and predation by animals and plants. And the new toxins he has discovered are being used as tools for improving our understanding of the human body and designing new and better treatments for certain diseases such as diabetes and chronic pain. Sam will share how his research can gain from pain. Most venomous animals will use their venoms defensively, and most stings or bites cause pain, but not all stings are equal. For example, the sting from a bee feels different from the sting of a scorpion. And Sam has had some firsthand experience in being stung in the name of research, and I know that you'll love hearing about it. Welcome, Sam.
All right. Hello, everyone. Thanks for coming along, and thank you to those online for coming, spending some time to listen today. So yeah, I'll be talking about how we can gain from painful stings and how we can use these to understand pain better and therefore come up with better treatments. So first of all, this is a Meet the Researcher event. So here's a little bit about me. My name's Sam Robinson. I'm an NHMRC Emerging Leadership Fellow. I'm also a National Geographic Explorer, and what that means really is those are who sponsor me and what I do. I'm a biologist, and more specifically, I'm a toxinologist. So I study toxins. And one of the cool things about what I do is I get to spend not too much time, but a little bit of time out in the field. For example, here there's a picture of me in the rainforest in South America, and you can see I have a pruning saw, and there's a wasp nest up there. So it didn't end as badly as you might imagine, but it was in the name of science. So you might be wondering, what does a pruning saw and wasps have to do with pain, and helping pain rather than just causing pain? And hopefully I can explain that to you here. So my research program is really based on some very simple observations. The first of these is that almost all venomous animals will use their venom defensively. And you think about the last time you stepped on a bee or got stung by, had a picnic and got stung by an ant or something like that. We all know this simple observation. And from this we can get this hypothesis that defensive toxins probably exist in some venoms. Second observation, which again many of us are probably familiar with, is that many stings cause pain, and sometimes it's pretty bad, right? So that leads us to develop this other hypothesis that these toxins are probably targeting our pain pathways. So I'll just explain that in a little bit more detail here. So pain signaling, maybe some of you know this very well, but some of you, this might be new to. When we experience something in our periphery, maybe it's painful, or for example, we touch something hot, or we smash our finger with a hammer. That's what this red lightning bolt represents. What happens is a nerve then sends a signal up to our brain, and we experience that pain actually in our brain. This is how pain signaling works. And different types of stimuli will cause a different signal to be sent, or they'll make use of different pain pathways to send a signal. Now, the third observation that really sort of underpins my research is this one, and that's that not all stings are equal. And I'll forgive you for perhaps not knowing this one. There's probably only a few people out there that have been stung by as many things as I have, and one of these people is Dr. Justin Schmidt, and he developed what's called the Schmidt Pain Index. So basically what he did was he rated different insect stings. He gave them a score of how painful they were. And as you can see, this picture really nicely shows it. The insects that are shown large here are the ones with the most painful stings, and things with shown small are things with less painful stings. And what else he did was he described those stings. So he describes, for example, the sting of a sweat bee. He describes as light and ephemeral, almost fruity. A tiny spark has singed a single hair on your arm. In contrast, he describes the sting of a bullet ant as pure, intense, brilliant pain, like walking over flaming charcoal with a 3-inch nail embedded in your heel. So that aside, it's fairly obvious there are differences between these two stings, and there are differences between a lot of stings. And that can lead us to hypothesize that venoms are almost certainly a rich and diverse source of pain-causing toxins. So what can we do with that information? This is where I come back to the lab and start the research. The first step, obviously, is acquiring a stinging animal or plant. And then the first question I ask is, what are the responsible pain-causing toxins? The second question is, how do they work? And the third question is, what can they tell us about pain? So Hana touched on this briefly before. Pain is complex. There are different types of pain. Hana talked about chemotherapy-induced pain, but there are many different types of pain. And we're using basically just a handful of ways to treat it at the moment. We need some out-of-the-box ideas, and we need to learn more about how pain works, or different types of pain work at a molecular level. So I hope you can appreciate that this is a sort of an out-of-the-box way to come up with new treatments. So the first step, acquiring some stinging things, turns out to be quite easy, particularly in Australia. So here's just an example of a few of the things that I've looked at, from scorpions, centipedes, wasps, some very big wasps, to nettles, to nettle caterpillars, to ants. And I'm just going to cover a couple of these today and what I've found from them. So one, if you're from Brisbane, you'll almost certainly be familiar with green ants, but you may not know what I'm about to tell you about them. So this one was easy to acquire. I've shown this little picture here. I walked across the street to the cricket oval and got myself some green ants. That's how easy it was to get hold of these. And if you've been stung, the pain can be pretty bad. It's sort of can be pretty severe, long lasting. But also if you get a really bad sting, it's accompanied by some really weird symptoms, including sort of local goosebumps and sweating. So this was a good candidate to think, oh, there might be something interesting in this sting. So one of the early steps was to collect some of the venom in order to study it. And this is how we do it. This is just a thin layer of waxy plastic. And you can see as the ant is stinging it, and it's quite happy to sting, as you'll see, we get these little droplets of venom collecting on there. Now, once I've collected those, I can analyze that, and I'll spare you the details here, but we use an instrument called a mass spectrometer, and we can basically take that little drop of colorless solution, separate it out, and each of these peaks here represent an individual toxin in that venom. We couple that with information. We collect the venom glands. Now, these are very, very tiny. If you look at that scale bar, that's 0.5 millimeter there. We take those, and these are the organs producing the toxins. And from that, we can extract the DNA, and we can build a library of toxin sequences. And with those 2 bits of information, we can figure out what's exactly in that venom. We can make those toxins, and we can test them, and we can figure out which ones are contributing to the pain behavior. In this case, it was really easy. It was a toxin called RM4A was the standout. So next, I wanted to understand how that was working. And to do that, I can test it on nerve cells or pain sensing cells. And what you can see here is I've set up the experiment in such a way that when the cells light up, I know that they've been activated by the toxin. And the pattern by which the cells light up tells us about what the specific molecular target of that toxin is likely to be. In the case of the green ant toxin, we found that it was targeting something called a sodium channel. And sodium channels, these are like little gates that separate the outside and the inside of our nerves, and they open briefly, and sodium ions flow through, and that's part of how our neurons signal. But what happens when you see that, we can measure that current of ions flowing through, and that's this little black spike here. So you see that lasts about 1 millisecond, that little spike normally. But when you have the green ant toxin there, it's like having a wedge in that gate that locks it open. And you can see that the current just keeps on flowing through. And it keeps doing that for, more than 30 minutes. So that sort of explains why we get this long-lasting pain. When you look at that on a bigger level, and those sodium channels, they're found in our nerves. They're really critical to the function of our nerves. And when the green ant toxin binds to those, it sends the signal up there, and we experience this long-lasting pain. So the green ant toxin, it's new to science. It's never been seen before. It's something unique to ants, green ants and other ants. And it's doing something to sodium channels and to our neurons that's never been seen before by science, and it causes this unique type of pain. It's a good example of something that we can use now as a tool to understand how those sodium channels are contributing to certain types of pain. And I'm just going to give a couple more examples of things that I've looked at. So one is bull ants. Again, probably most of you are familiar with bull ants. They can give a pretty nasty, short-lasting sting, but they can also cause hypersensitivity. So if you get stung a few times, say on your arm, and you get stung again a day or so later, that site will actually be hypersensitive to those stings. The other thing is the Australian stinging trees, or gympie. These can cause pretty severe pain, reasonably long lasting, so a few hours. But one of the really interesting things about the Australian stinging tree stings is that once the pain has subsided after a few hours, you can bring it right back by just getting it cold again. And that's a phenomenon called cold allodynia. And that can last for weeks, even months sometimes. So it's this really unusual type of pain. So the green ant, as I've shown you before, targets something called a sodium channel in our nerves. Turns out the bull ant there's a toxin in bull ant venom that targets something completely different, and that's known as an EGF receptor. And what that does is actually sensitizes our nerves. It makes them more sensitive to normal stimulus. So it hurts more when we experience something painful. And the stinging tree actually has a toxin in there that targets something called TMEM233, and you don't need to remember that name, just that it's you see it's this funny sort of code. That's because it's never been characterized before. No one's identified what this protein does or how it contributes to anything in our bodies. And now from our research, we know that it's a key player in certain types of pain, particularly that cold sensitivity. So what we've found, just from these 3 examples I've given you, we've come up with 3 new classes of toxins, which are able to cause 3 different types of pain. And we've been able to identify new pain targets. And just to summarize, I hope that I've convinced you that venoms are not just a pain. They can actually teach us about how pain works at a molecular level. And this information is really, really valuable. We can identify new pain targets. We can find things that we didn't know were involved in pain. And if we can do that, we can come up with new treatments, completely new classes of painkiller. Instead of whipping the same old dead horse, using the same types of drugs, we can actually come up with completely new classes of drugs based on this information. And if we have new classes of drugs, we're going to have new treatments for different types of pain. And thank you for listening. I believe questions are afterwards, so I'll do that later. Thank you.
Thanks so much, Sam, and thank you for taking one for the team and getting stung so we don't have to. I'm pleased to introduce Dr. Fernanda Cardoso, who's an expert in biodiscovery and drug development and an emerging leader in studies of animal venoms applied to novel drugs. Dr. Cardoso explores the amazing world of venom-derived bioactive molecules and to study their effects on human physiology with applications in neurologic disorders such as chronic pain and neurodegeneration. Today, Fernanda will explain how her research in drug discovery has shown spider venoms are rich in components with powerful analgesic properties for the treatment of chronic pain. These new analgesics are a novel type of drug with the potential to provide better safety and pain relief and a promising alternative to opioids. Welcome, Fernanda.
Thank you very much, Wendy, and hello, everyone. So my name is Fernanda Cardoso. I'm originally from Brazil, so different from Hana. I made a very easy transition to Brisbane, no problems at all, and the Australians are as laid back as us as well, so very pleased I'm here. And so today I'm going to talk a little bit about our program in development of novel analgesics. And different from Sam, where he's talking about venoms that induce pain, I'm going to talk about venoms that are actually doing the opposite. They are actually blocking pain. So why are we interested in venoms? We actually have a history of venoms being applied in the development of new drugs. The first drug derived from venom, it is a drug called captopril. For a coincidence, it is from a pit viper Brazilian, and this actually has really sparked the interest of Brazilians to be studying venoms for drug development. And this drug, it's used for hypertension and is the most popular drug in the market for this condition. It's used worldwide. So this was a big mark for researchers to understand that venoms actually have a great potential. It's a great starting point for looking for new drugs. So after this mark, we had other drugs from snakes as well, some drugs that are assist in the coagulation cascade or can help with hemorrhage, and also for diabetes type 2. But the most important one I want to talk about today, which is linked with our work, is this drug called Prialt. So this is the first venom-derived drug used for chronic pain. It is used in severe cases, cancer pain, or other types of pain which are resistant to opioids. So this was a big mark for us. And because this drug has a very similar properties with the molecules we are interested on, so these make us quite confident that what we are working towards now is going to become a drug in the near future. So I'm also very interested in voltage-gated sodium channels, like Sen presented earlier. So we have a number of animals that produce venoms that target these particular channels, mostly because they are in pain pathways, and these drugs, they will be activating the channels, causing pain. But in the mix of cocktail of venom molecules, we also have drugs that will be blocking these channels. And these are the ones I'm interested on. So we have, for example, here a few examples of animals that will be targeting this is a white channel, and we have in yellow a picture of how a drug is going to be sitting on the top of this channel, which will be inducing the blockage of the ion flux. I'm going to talk a little bit more about it. And what is interesting is that although they are all targeting the same channel, they have different strategies. This channel is very complex, has different binding sites, which means that, for example, puffer fish toxin, the tototoxin, and Conn's neurotoxins, they like to target the pore, which will be this area. But spiders, they like to target a different area of this channel, which is in yellow here. This is a drawing of a spider peptide on the channel. And this makes these venoms special and better than the other ones when it's to develop analgesic. I'm going to explain you why. Here, just have an example of a 3D structure. I'm not going to go too much details about the structure of these molecules, but what I want to highlight is that they are very resistant, very stable, which is a big advantage for us. So they naturally evolved this very resistant structure where changes in pH or other agents will not be able to destroy the activity of this molecule. And sodium channels, as Sen was saying before, they are very important molecules in our physiology, but not just in pain. We have sodium channels involved in a number of other physiological events, including heartbeat, secretion of hormones. We use sodium channels to move our skeletal muscles. And we know that sodium channels are also involved in some types of cancers. So these channels, they are very important targets for drug development, not just for pain, but for other conditions as well. And when I look at a little bit more detail about what type of conditions this could be, that could be taking advantage of molecules like the ones we have as novel therapeutics, we have here neuropathic pain, where we have a number of sodium channels. This NAV is the sodium channel. So I have a number of different subtypes: visceral pain, cancer, motor neuron disease, paralysis, some types of epilepsy, and again, cancer. So there is a number of conditions, complex diseases that actually can benefit from this library of molecules. I'm very interested in studying in detail the composition of these venoms. And I want to know what is in the venom, as well as what kind of ion channels these venoms are modulating. So we have developed here at IMB, with a large team of researchers, some discovery platforms where we'll be using different techniques. It's a multidisciplinary team where we can go from venom. We can go from venom, separate each molecule of the venom, and as well as evaluate the activity of each molecule against a range of targets and ion channels that are modulating pain pathways, not just sodium channels, but we can look at other ones as well. So with this platform, we have discovered, for example, even venoms that have been very studied, have been studied throughout many years by researchers around the world, because they haven't been through platforms like ours at the IMB, they haven't been underlooked. So we are revisiting lots of other venoms to find out that actually, even the most studied ones, we have still 65% of the content of peptides is still to be studied. So it is really an important resourceful cocktail of bioactive molecules for us to be exploring. And by doing this kind of work, we can look at diversity. So here I'm just showing the type of molecules that we will have in this unexplored pool. So we have different molecules with different pharmacological properties, and they all can have a different utility for our programs. And we can look at as well, which I'm not going to tell in details, how each part of the molecule is participating in the binding and modulation of the ion channels. And so I'll give you an example. So this was a project where we look at 28 spider venoms. Most of these spiders, they are from South America. There is a bias for large tarantulas in our projects, mostly because of access of venom and as well as access to the actual spider. So we have a good collaborator which would visit people around Europe and South America that actually have the spiders as pets and would milk the spiders. So we have around probably 500 different species of spider venoms in our library, which we can use right now to investigate for whatever type of receptors we are interested in. And once we select interesting peptides, we have automated platforms where we can look at for each individual type of ion channel. So this is a very detailed work to know exactly what that venom component can do to our body. And I'm giving you an example here of a few of the peptides we have isolated. So we have the tarantulas, and some of the peptides that have been studied, published, and some are also unpublished. So this is all ongoing work, and each of these molecules, DF1A, TAP1A, Protox3, they are already in preclinical levels of investigation, giving us great promising results. But today I'm going to talk about a specific type of pain, which is visceral pain. So we know that visceral pain, by the literature, is influenced by alterations in F1.1, sodium channels of type 1.1, 1.6, 1.7, and 1.9. So what did I do? I got back to my large library of venom peptides, and I started to search for the peptides that could have that particular property, pharmacological property, to target the main sodium channels, having an important role in the visceral pain. And in collaboration with great colleagues in Flinders University in South Australia, we have decided to test these peptides into the colon in a model that represents some types of IBS, for example, or as well as testing in a model for bladder disease. So we want to see if these peptides are able to revert chronic pain, particularly visceral pain. So from this spider Theraphosa hypothesis, which is found in Brazil and Venezuela, we have isolated these two peptides, TAP1A and TAP2A. And again, I did a very detailed pharmacological profile of these molecules against sodium channels. And we found that TAP1A actually really hits the most important subtypes of sodium channels for us. And I was like, okay, I think we have a good candidate here. And I think we have a chance to actually extend these studies from in vitro to more preclinical. So now we want to see how this will how this affects the bladder physiology as well as bladder during a pain condition. And we can see here that when we test this peptide intravesically in the bladder, that TAP1A can revert the pain sensation. And we can see here, these are just recordings of spikes of sensory neurons in the bladder. And we can see in the presence of increasing concentrations of TAP1A, this neuronal hyperactivity is reverted. So this was quite exciting for us. And we can see here, this is the control for muscle contraction in the bladder. So the bladder is actually safe for their physiological functions. It's just targeting the neurons. And this is a very important aspect for us. And we also test that in a colon, in a colonic preparation, because we are also interested in testing the ability of this peptide to reverse pain related to irritable bowel syndrome. And we can see here that in the presence of the peptide, the venom peptide, we have a decrease in the signal, and we can see here statistically significant. So this was enough for us to make a decision that this peptide is showing promising modulatory activity for reducing pain, and we want to move to a preclinical model. So we chose a preclinical model of chronic visceral pain, which mimics visceral pain in IBS. And we learned that in the presence, so I'll just explain you briefly, this is a health control where stimulus of pain goes as expected when we are applying a pressure in the column at around 80 milligram, about 80, we have the normal pain, but the animal that is suffering from IBS has a much more sensitive column and it starts to feel pain way earlier. So it's a hypersensitive column. And that when we applied the peptide, the venom peptide, we could revert that to normal pain sensation. So that was quite promising for us. And here just showing the statistic significance of our work, which we're quite pleased about. So we learned that this venom peptide, it is an ideal drug lead. It has great pharmacological properties. It has proven ex vivo and in vivo effects, and it has a great feasibility of production in chemical synthesis. So we have something that has a preclinical therapeutic effect and that we can actually produce in large scale. So with this in hands, we are looking at options of how can we make these venom peptides better. So thanks to technology, the information available for us researchers in the literature is really getting rich and useful for us to transfer knowledge from other research in venom peptides to our own research in such a way we can make our peptides better in terms of how they target the sodium channels, as well as how selective they are. We don't want peptides that are going to target the sodium channels in the muscle. We do not want peptides that are going to target sodium channels in the heart. And we need to keep that in mind all the times. So we can improve our peptides for improved inhibition as well as selectivity. So we'll try to apply that for this candidate, TAP1A. So I just created a strategy that I call OPT strategy, optimization strategy, where I transfer pharmacological characteristics from one venom peptide to the other until I reach one common optimized peptide, where we can test in vitro to test the pharmacology as well as in vivo. And that's what was the most exciting part. We saw that the TAP1A wild type, like we isolated from nature, it can revert pain in vivo. We can see here compared to the control, but the optimized one has a way more potent activity. So this means to us that we are actually understanding the mechanism now. We understand how these peptides work, and we understand how we can optimize them. And this is very important moving forward, so we can make these peptides more useful and safer for us. So just to finalize the science part, during this journey, we really learned how these peptides bind to the channel. And I'll give a brief explanation. I think it's fantastic. These channels, they are voltage sensors. They're voltage sensitive, which means that if there is a change in voltage inside the cell, the channel changes conformation. And when it changes conformation, there is one particular part of the channel that moves up, and then the pores open and the ions go inside. The spider toxins, they bind on the top of this particular structure. So when the cells try to depolarize, the channel is locked and cannot open the pore. And this is a very conserved mechanism, not just for our peptide, but if you look at other studies, it is spread around many other peptides from spiders, particularly from spiders. So spiders are definitely one of the most amazing source of molecules for the development of novel drugs, which are safer and more efficient. And what would be our clinical strategy to apply these novel venom peptides? So I gave initially the example of Prealt, which is the cone snail venom peptide who is in the market, used for the treatment of chronic pain. It is a unique alternative for opioids. It belongs to the same class of venom peptides as TEP1A. It is also an ion channel inhibitor, although it inhibits a different class of ion channels, not the sodium channels. In this case, Prealt inhibits the calcium channels. And our drug administration strategy will expand on the knowledge built on the Prealt. What does it mean? TEP1A, for the treatment of IBS, we envisage it being administered into the colon, a gut-restricted drug, so it's not going to leak into the system. And for the bladder conditions, we see TEP1A being administered intravesically, which will also impede that the peptide will spread into the system and guarantee that side effects would not be a problem. So, of course, this work will not be able without the contribution of many, many amazing scientists, in particular, Professor Richard Lewis, Glenn King, and Arina Vetter here at IMB, Flinders University, Stewart and his team, also Volker, who has the amazing libraries of venoms, and as well as Professor Marco Vinicius back in Brazil. So, and of course, the funding agents. So this work has been mostly funded by the NHMRC in Australia. Thank you very much.
Thanks so much, Fernanda. And I hope that you enjoyed that. As much as I did, I'm still on an extremely steep learning curve, having had my last formal science study in the 1980s. But the team here has been really generous in sharing their knowledge and helping me along the journey to understand how we harness nature to find new cures. We're privileged to have Nicolette Ellis from Chronic Pain Australia, and she's agreed to join us today to share some perspectives from the community. Chronic Pain Australia is a national voice of Australians living with chronic pain. Chronic Pain Australia hosts Australia's largest pain awareness campaign, National Pain Week, which will be 24th to 30th of July. And during National Pain Week, CPA shines a light on the lived experience of chronic pain through the National Pain Survey, Australia's largest annual survey investigating the lives of people with chronic pain. Nicolette's drive for advocacy and meaningful change comes from her own personal experience with chronic pain and is a core reason she's dedicated her career as a pharmacist specializing in chronic pain. Welcome, Nicola. That's all right.
Well, it's wonderful to be here and to listen from the researchers here today around the types of novel treatments that are coming into the chronic pain management space. As Wendy had mentioned, I'm the president from Chronic Pain Australia. We're the national voice for Australians who live with chronic pain. So there's 3.6 million Australians who live with chronic pain. And I think if you were putting your bets on these researchers, they probably develop a blockbuster drug, what we call a blockbuster drug. So in the pharmacy world, a blockbuster drug is something that, you know, has huge sales like atorvastatin or I'm a pharmacist and I've lost all my drug names. So, and I think in chronic pain in particular, we don't have any really novel targeted therapies in the medicines pipeline. And it's brilliant to see what Fernando, Sam and Hana are doing in this space. And I'm really sick of being a pharmacist in this space because a lot of the time I'm, you know, using Sam's words, beating that horse with medications that have not been developed and not targeted for these different types of pain conditions. So this type of research is so important. And Hannah really got me at the beginning because I've worked with so many patients who have survived cancer, but now living a total new world, which is not a fun world. And unfortunately, many of them say to me that, you know, I wish I didn't survive from the cancer because this pain is just all encompassing and it's taking over my life. So I really think we need to put a lot of work behind and funding behind researchers in this space. If you look at all of the chronic disease states, asthma, diabetes, mental health, cardiovascular conditions, we have drugs that are very targeted in that space. And then when you look at chronic pain, we really don't have very much in that space at all. So we're part of the National Pain Alliance as well, which is to lobby the federal government. We're the consumer organisation on that board to level the federal government to put some more investment, a solid investment into chronic pain and research and novel medicines behind that. So wish us luck on that journey and please become a member with Chronic Pain Australia. We are advocating for the rights, the lived experience of people with chronic pain and their carers. And join me on the 24th of July for National Pain Week, which is our largest week where we talk about chronic pain management, and hopefully we might see some of these wonderful researchers there as well. Thank you.
Thank you so much. I really appreciate you joining us, and I'm pleased now to move to the Q&A session. And can I please ask our panelists to all come join us up front? And for those of you online, on the bottom of your screen, you'll see a Q&A tab to type in your questions to our researcher, and SJ will help to facilitate that. So can I ask anyone with questions? Just identify yourself and let me get the mic to you so our online audience can also hear from you.
Thank you. I don't know who to address this to, but have you done any research on phantom limb pain?
No, we haven't yet, but I'm pretty sure that it's something that if we have major interest in pursuing, that we can start to look into implementing some screening assays for targets or even preclinical models that could help us to find out if our drugs would be useful for that type of pain as well. At the moment, I don't think there is anyone at IMB doing that, but definitely an interesting field, and we can check if our molecules could be useful for that.
I'll just add that we are looking at the peripheral nervous system, and that is what is affected in phantom limb disorder. So the compounds that we're studying will be relevant in that for sure.
Thank you. Many of these novel treatments are peptides or proteins, so presumably they would have to be injected rather than taken orally. I was just reading an article about pharmaceutical companies using mRNA to make protein enzymes in the body. Is that a possibility that you're looking at?
Definitely yes, because we work with proteins and peptides, the mRNA system, it's quite applicable to our research. So it would be just a different delivery system for us. But we are looking into it. Yeah, we can code our venom peptides using mRNA, which is fantastic. Yeah.
So I have looked not directly into that, but we do have, we've tested some compounds that if you deliver them with RNA, you will only, or as RNA, they'll only be expressed in certain tissues. So say the affected tissue, and the example we've been testing in is arthritic, the synovial fluid of arthritic joints. So if you can express a peptide or something that's analgesic only in that space, you're going to limit the side effects. So yeah, we have been looking a bit at that.
I have two questions from Jo online. The first can be answered by any of you if you have any information. She just said, any thoughts on complex regional pain syndrome and pain pathways and targets with this condition? And the second question is actually for Sam, and it's just about bee venom and if you've investigated bee venom and its effects.
So I can comment on the complex regional pain syndrome. This is a condition that is often seen in children, and basically it includes severe pain all over the body that is impossible to treat. So basically there are currently no treatments to this, but we are slowly starting to understand that actually immune system plays a really important role in the development of the complex regional pain syndrome. And for example, using some anti-inflammatory drugs that basically target immune system can actually help those patients basically in recovering and preventing pain.
Yeah, so I'll answer the second question, which was, have I looked into bee venom and how that is causing pain or treating pain? Was it? Have I looked into bee venom? I have looked into bee venom. I've actually found out just last year why bee venom or bee stings hurt. So the chemistry behind that. Would you believe that that took until 2022 to figure out? I know that some people, there's bee venom therapy has sort of become popular with certain arthritic conditions. Based on what I know, I wouldn't what's the word, recommend it. But you know, sometimes your suffering's really bad and you just need something. So I won't say too much about that. Right.
Have you done any studies on neural sensitization disorders as they affect chronic pain?
I think a large number of the neuropathic conditions we studied, they involve hypersensitive neurons. So I think this is one of the most common characteristic among different types of pain. The neurons, they are hypersensitive, which means that they will be firing in sensations that should not be causing pain, but they will fire as there is a pain signal. And that's what we would call hypersensitive.
Yeah, and I'll add that a lot of these hypersensitive syndromes, they don't have good treatments, and some of them don't even have treatments at all. And you might recall from my talk, we've actually identified right here in the IMB, this building, 2 new potential targets to treat different hypersensitive disorders, and both of these came from pain-causing venom. So one from the bull ant, which is our Australian-owned animal, and the other is the Australian stinging tree. So that's told us, given us a potential pain target for this cold allodynia type of hypersensitivity.
I don't know if this is relevant here, but I know several people who suffer from peripheral neuropathy, where you can get sensitized, I suppose, to, I think it's something to do with the myelin sheath on the nerve degenerating from the extremities upwards, usually feet and then eventually hands. And a lot of people suffer from this, diabetic people, but also others, like my husband. And there is nothing you can take whatsoever. Does any of your work, will that be relevant in this?
So yes, neuropathies are often caused, as you said, and it has to something to do that when the neurons basically lose myelin sheaths, and this happens often with the largest and longest neurons. That's why this type of neuropathies first develop in feet, because basically the sciatic nerve that goes all the way down, it's about a meter long. So basically those nerves are very long and they are impacted most. And as you said, there's nothing there for the treatment. And as I mentioned in my talk, basically those patients that are treated with cancer therapies, they often develop this type of neuropathies. And so we are looking closely into the mechanisms, but as I said, the mechanisms are very, very complex, because it's not just the condition, like for example, diabetes, or it's not just the cancer treatment that causes those neuropathies, but there are all the other factors like gender, age, and also other basically comorbidities, like those patients, especially older cancer patients, they will have other diseases that contribute to this as well. So we need to look at this as a complex, really this complex system, and we need to look how also the nerves in the body interact with other cells, because we often tend to look only at the nerves and at the peripheral nervous system, but we also know that other cell types like immune cells or such as immune cells, they play important role in the development of these neuropathies. And as you said, there are no treatments. There's nothing we can use. So we can try to help those patients short term using opioids, but opioids have really side effects and they actually do not really work. There have been some attempts to perform clinical studies using, for example, antidepressant, tricyclic antidepressive drugs, or drugs that we use for treatment of epilepsies, but these type of drugs, they show only little effect, and usually those patients have to suffer, and there's nothing we can do currently. So that's where we are trying to put our efforts in understanding the mechanisms, so that we can actually basically interfere in the pathway of the development of the neuropathies somewhere, and basically prevent them even from happening.
We probably have time for one or two more questions. And I just wanted to mention, we've had a couple of quite detailed questions online that we would be happy to respond via email, just in the interest of time. And we'll please, SJ, who's currently facilitating online, will provide details of how to email in. Does anyone else have any questions in the room? Thank you so much. It's been a really fascinating session. I hope you've enjoyed it as much as I have. We're coming close to the end this afternoon, but we just wanted to share with you a couple of ways that you can help. You may have heard from a couple of us today that there are definitely challenges that are holding research back. Government funding is not a growth industry. And so we work with a range of partners, and we really appreciate all of the supporters who do come along. Here are some ways that you can help our researchers. Follow us on social media. If you're not connected, we're happy to help you. Sign up for our online newsletter. We send out an informative newsletter, I think every month, is it, SJ? Every month, yeah. And that's a great source of all the latest in our research. Soils for Science is our citizen science program, and that's an opportunity to participate and also contribute to research on antimicrobial resistance. Encourage corporations and community groups to organize a free tour or a guest speaker. We love sharing what we do. IMB is one of Queensland's best kept secrets, and we prefer not to stay that way. Any donations go 100% goes to research, and in fact, that also triggers an additional 30% that helps pay for the on-costs of research. So it's more than 100%. And if anyone's interested, we'd love to welcome more people into our director's circle. That's donors who donate more than $1,000 or more per annum are members of our director's circle, and that's a really special group who we welcome along to lunch with our director each year and a number of special events throughout the year, and we'd love to have more of you along. And now this is, as you may have guessed, my first time emceeing. SJ usually does this, but I said, please let me try because I'm new. So please bear with me. We just have a bit of a video sharing a little bit about the Director's Circle.
Scientific discovery and collaboration are the key to a better future. At the Institute for Molecular Bioscience, more than 500 staff work across disciplines and through extensive international networks in order to improve outcomes in health, agriculture, and sustainable futures.
The contributions of our philanthropic donors allows us to continuously advance our work improving outcomes in health and disease and to finding sustainable solutions for our cities, fuels and foods.
Our research is inspired by nature. We maintain the largest collection of venoms in the world, which we use to develop environmentally friendly insecticides and life-saving human therapeutics.
I feel enormously privileged to be part of the Director's Circle. This wonderful group of philanthropists are funding the next bout of amazing scientific ideas and progress, and it's really wonderful to be a part of that and to be so closely aligned and enhancing the outcomes of the Institute.
So last year, thanks to funding, I was able to present my research at an international, highly regarded conference. At the conference, I was able to not just present my research, but get feedback from international researchers from all around the world and forge new collaborations. And this has really been key to my career development. And none of this would have been possible without the generosity of my donors.
We do have friends with serious health problems. If we can see some tangible results for them in our lifetime, then that's what we'd like to see. The whole purpose of our contribution is to help society. By helping IMB, we can help society.
Special interest would be into dementia. My mother had dementia. It's still there that you remember it and you can feel for other people who also are facing those problems with their parents.
Through philanthropic donations such as the Ultrabar Alzheimer's Researcher Program, I was able to pursue my PhD over here in IMB, the leading research institute in Australia, and was able to study Alzheimer's disease and inflammation.
I hope the impact of my philanthropy is twofold. Firstly, I want to see fantastic science at IMB performed, really blue sky research that's not able to be funded through the traditional mechanisms. Two, I want to see all of the fabulous women at IMB succeed in their career goals and to overcome the barriers to their career progression and prosper so that we have eventually full participation of women at the senior levels at IMB.
Well, fortunately, we have met some of the researchers involved, and it's always great to meet those people who are doing that fantastic work. And just as fortunately, we have met some of the families of people who have benefited from the research we have supported in the past. So it's a real privilege and quite humbling to just step inside their lives for a couple of minutes and understand how important this research can be to their lives.
Funding from private donors, UQ alumni and the lot has greatly accelerated our research aimed at developing game-changing drugs to treat stroke and heart attack and to increase the pool of donor hearts for transplantation.
We were awarded $10,000 through the disbursement of funds from the Nobel Prize in cryo-electron microscopy. This work allowed us to produce a first novel therapeutic in microalgae. designed to support prawn farmers whose industry had been decimated by a white spot virus outbreak. This funding allowed us to produce the protein and conduct the first field trials.
Members of the Directors Circle play a significant role in the critically urgent work of IMB. Your contribution lights the way to our next big discovery.
Thank you all for your wonderful questions today. As we begin to wrap up the formalities, I'd like you to know that we do, of course, plan on bringing you more of these Meet the Researcher programs. And we kindly ask for your feedback on the presentations that you saw today. As you give this feedback, you can choose to sign up to our newsletter or stay in touch with the exciting progress being made at IMB. Get in touch. My colleague Ken Lai at the back, give us a wave, Ken. And I were always happy to have a coffee and a chat and to share updates on specific areas of interest to you. So please do not hesitate to reach out. And thank you so much for your time today. We're grateful for your interest in IMB's work, and we hope to see you again soon. Thank you.